for an introduction. It's a pleasure to be here and listen to all the awesome talks being presented at this conference. Give you a little bit of context. This work was done at the Simons Foundation, a nonprofit research institute. And since then, I have spun out my own startup AI company that's focused on leveraging our findings and bringing them into translation. For those of you that are not familiar with autism, autism is on the rise. It's been exponentially increasing since the 1970s and affecting 1 out of every 44 children in the US. It's a heterogeneous cognitive, behavioral, and communication disorder that is where every child is very different in terms of the symptoms that they have. And that about half of these children have issues with their GI symptoms that has been noticed since the 1950s. And underlying mechanism has been largely unknown, but over the last decade, there have been numerous microbiome studies that have attempted to link the autism symptoms to the microbiome. But there's a lack of consensus on exactly what is driving this connection. And so what we did is we conducted the largest meta-analysis up to date, where we looked at all of the autism microbiome studies that we can get our hands on, as well as all of the adjacent omics levels to get a better understanding of what exactly is driving could be driving the autism microbiome connection. Starting with the microbiome, microbial enzymes extracted from shotgun metagenomics, the microbial metabolome, the virome, human genome, human transcriptome metabolome, and as well as human host immunity. Altogether, we looked at 18 studies spanning 26 data sets. And one thing we realized as we were putting together these data sets is that in order to properly analyze these data sets together, we need to revisit the fundamentals of doing proper statistical analysis. For those of you that are familiar with these data sets, you might know that most of these omics data sets consist of feature counts that are both sparse and compositional. What I mean by compositional, I mean that oftentimes the absolute concentrations are not available, which turns out to be a major confounder when you are running differential abundance analysis. But one thing that was in our favor is that most of these data sets were well designed, meaning that there was they were age-sex matched, which is important because in autism, males are more likely to be diagnosed with autism compared to females. But as many of you know, the batch effects are a persistent problem with these types of analyses. Folks use different types of sequencing technologies, different extraction technologies, all of which are confounders that hamper reproducibility when analyzing these data sets. But with this in mind, we saw this as an opportunity for novel methods development to enlist the powers of Bayesian uncertainty estimation and revisit how we do differential abundance analysis to facilitate this type of meta-analysis. What I mean by differential abundance analysis, consider this toy example. Here we have a toy environment with 12 orange, 12 blue microbes. As the experimentalist, we can only observe a subset of these microbes highlighted in the dotted line here. And suppose that we have the question, we have a perturbation of this microbiome, and we want to ask ourselves, what exactly has changed? In this particular case, you might be tempted to say that the orange microbes decrease in abundance. But the reality is that we can't say for sure because there's an equally valid scenario where using the same sequencing data, we cannot rule out the scenario where that the microbial load has increased in abundance. The problem here is that we don't have the access to the absolute counts, and thus without this data, we cannot differentiate between these two scenarios. So unfortunately, all the microbiome autism data sets that we collected do not have absolute quantification performed. So what can we do? So we came up with a method back in 2019 that proposed a concept of differential ranking. So the idea given is that when I want to infer a log fold change, let me first consider the log-fold change in absolute terms. I can take this term and I can split it into two parts. The first part here being the log-fold change that I can directly infer from the sequencing data. And then highlighted in red is a log-fold change that is due to the change in the total number of cells before and after this perturbation. What that basically means is that when I'm trying to interpret my log-fold changes pulled out of the standard differential abundance methods, I actually don't know where 0 is. And as a result, I can't tell you if microbes are increasing or decreasing abundance without making some assumptions about the underlying system. To get around this this limitation, what we realize is that the change in absolute abundance is just a single term that's equally applied to every microbe. So what we can do is instead of asking if microbes have changed, we can ask which microbe has increased the most, which microbe has decreased the most. We can always sort the microbes based off their log-fold changes. And that turns out to be consistent between the absolute abundances and the relative abundances. Of course, that's just one of many problems when dealing with these types of meta-analyses. Just to give you a hint, there's certainly batch effects. Now, this is a reanalysis of the MBQC data set where they took multiple samples and sent it to various labs to reprocess and to see, well, how prevalent are these batch effects? Well, on the plot on the left, you can see that if you just take two samples, that indeed you can sort of tweeze out the differences, that they are two distinct samples. But then you can also see on the right that there's definitely batch effect. You can uniquely identify which lab sequenced which samples. And but it turns out that the batch effects problem can be somewhat alleviated when you consider only the differences within a lab. So when you run your differential abundance approach within lab and compute the log-fold changes, turns out those log-fold changes are actually consistent across the labs, suggesting that these batch effects are largely linear in this specific case. The implications here is that when we perform differential abundance per lab, we can basically aggregate the differences across the labs, which will allow us to build stronger consistency when we're doing the downstream analysis, when we're aggregating these data sets in our analysis. The last point I'm going to make is that this solves some problems, but not all the batch effects, especially when we want to combine the shotgun metagenomics data with the 16S. And so here we can see that when you do just high-level cluster, even though these 16S and shotgun metagenomic samples are collected on the same samples, they will strongly separate when you try to cluster them together. So this is why I'm teamed up with Chi Yun and Daniel McDonald, and together we came up with a new method called Green Genes 2 that builds a universal phylogeny that unifies 16S and shotgun metagenomics data. And so here we can see that when we compare the raw abundances, they're strongly consistent at the genus level. You have much greater consistency with the log-fold changes, and that the batch effects largely is that giving us hope, confidence that we can combine these data sets together. Altogether, with these building blocks in place, we can build a new differential abundance model that can account for batch effects, that can account for various experimental designs specific for the autism meta-analysis. So here we designed a Bayesian model that leverages the negative binomial distribution to account for the tremendous sparsity in these data sets. That allows us to control for sequencing depth, negating the need for rarefaction while accounting for the issues surrounding compositionality, while allowing us to infer the differences between ASD children and their neurotypical age, sex matched counterparts, allowing us to account for age-sex confounding. And overall, we do see that using this approach, there is a strong signal that there are microbes that are strongly associated with autism that do differentiate the autistic individuals from the neurotypical controls. is so strong that you can construct simple diagnostics that not only differentiate these kids across data sets, but you can even do cross-validation with them. That this generalizes to shotgun metagenomics data, which is a completely different data set from the training that was used to extract the biomarkers. We can then do a comparative genomics analysis. We can take these microbes and pull out the genes that differentiate the bugs as strongly associated with ASD from those commonly found in the controls. And then we can even cross-reference them against pathways that are differentially expressed in post-mortem brain tissue. What we can see here is that there's evidence that there is strong cross-linking between human brain pathways and gut microbial pathways, up to 95 potentially cross-linked pathways. And that there's a strong overlap in pathways implicated in amino acid biosynthesis, vitamin metabolism, mineral absorption, and that this overlap is also seen when looking at the deficient dietary preferences in autistic children, that they tend to be deficient in amino acids, vitamins, and minerals. And we can also take the microbes that we identified to be associated with autistic children and cross-reference it against an FMT study. In this particular case, authors showed that when you apply FMT, you're seeing about an 83% efficacy rate 2 years after the FMT was applied, meaning that the kids' motor symptoms improved, their GI symptoms disappeared, and their behavior symptoms were largely reduced 2 years after the FMT. And so when we cross-reference the microbes that we deem to be associated with ASD, 91% of the bugs that we identify were knocked out post-FMT. To wrap this up, the associations between the microbiome and autism are real and strong from what we've seen, and that the associations between the microbiome and other omics levels hint that there's real functional crosstalk between the host and the microbial systems in the context of autism. The fecal matter transplant provides an initial hint at causality, but more work is done to identify the mechanism of action underlying FMTs. And with that, I want to acknowledge that this was a tremendous effort that started out at the Simons Foundation. And I want to acknowledge Jim Simons for his philanthropic contributions for making this work happen. He, as many of you might have known, he passed away just a few months ago. And this work could not have been done without his contributions. And with that, I'd like to thank you for listening, and I'll take any questions.
Time for one quick question.
Great talk, Jamie. So it's really fascinating that across the studies you're seeing this common pattern, but when you were showing the list of studies, there was like a subset that it wasn't predictive of. And as you were saying, it's such a spectrum of disorders. And I'm just wondering if those studies varied in, for instance, whether they specifically selected people who had comorbid gastrointestinal symptoms or food, kind of restrictions or things like that.
Yeah, so there's only so much I can squeeze in a 10-minute talk. Longer versions do address this. So a couple of things on this. First off is that when it comes to GI symptoms, the effect size was the strongest for studies that selected for kids with GI symptoms. The FMT study I discussed was solely focused on recruiting kids with GI symptoms. The second part of that is that when we looked at studies that were sibling matched, we didn't see a signal. That is very mysterious to us. We're not clear why, but I think you cannot ignore the genetic component, that there's a significant subset of autism that has no genetic causes.